We're joining you from the lands of the Wurundjeri people of the Kulin Nation. We also pay our respects to elders past and present. And today we really want to kind of delve into a topic that both Katie and I find absolutely fascinating, and that we're very conscious is often, like so much in the stem cell space, misrepresented in pop culture, in the media. And it's something within the stem cell community that we're very conscious of. And I thought this was a great time to give you an insight from sort of within our community, the scientific community, and particularly with Katie's expertise, to really delve into where we're actually at. And to look at this question of whether stem cells and in vitro gametogenesis is actually the future of fertility. Now, this is an area I've been interested in for a long time. I started my career working in an IVF laboratory where I was a clinical embryologist. I made, embryos. I actually started way back in the late 80s when it was a very new technology. And in fact, at that time, we still didn't even routinely freeze embryos, let alone use intracytoplasmic sperm injection to help men with infertility achieve a family. So, I've both, I suppose, watched the progress of the field, but I've also been concerned about, as I mentioned, the misinformation, and some of the headlines I've gathered here, I think illustrate that. There's one at the top left, the mouse that has two dads, was from, I think, 2010, 2011. But every kind of development, scientific development in this field, as in so many other fields, is often accompanied by these headlines that have a lot of hope, or at least are read by people who are looking for help or for assistance in some way, and perhaps will further fuel their hope. So this is something that the kind of the area that Katie and I really want to expand on today, is go behind these headlines and talk about the science. And one of the sort of impetuses for getting together was that we both read this article that appeared in the New Yorker. It was entitled "The Future of Fertility: A New Crop of Biotech Startups Want to Revolutionize Human Reproduction." And we were both really struck by this article and the people the journalist spoke to, quite frankly. And I think it was some of the very similar issues that I've seen before around, I suppose, promissory futures. But this time it has a much stronger and very motivated biotech sector that appear to be behind it, who have raised substantial funds, in the order of, I think this company here, Conception Biosciences, have recently raised something like 40 million in a startup round. So, and I thought this quote at the beginning, that IVG is already seen as the future of reproductive science, was quite interesting. And the CEO here is reflecting on going to a recent scientific conference where she says that it was hinted that gestation that could happen outside the body was not a matter of when this will happen, but actually it's going to be happen, and how can we use it? And so IVG was sort of added into this additional reproductive technology. So I thought that was quite interesting how it was presented, and many other aspects of the paper are also interesting. But there was also this comment that I thought was worth sharing, that this issue of, if the technology was safe and effective, who's going to access it? And you can see here someone reflecting on the fact that it might be only parents who send their kids to private school or get the kids piano lessons or ballet lessons that can access it. So this idea, again, with so many other technologies, is will it be equitable access? But I suppose for us, one of the most significant questions is, what is the science, and does the science even justify these kind of headlines and this conversation? So what we're going to do today is to talk about the biology of gametes, gonads, and stem cell technologies. We're going to talk more about what IVG is, what has been achieved in laboratory in different species, and then what the challenges are, both from a scientific perspective, but also beyond the lab and what the implications are. So Katie's going to really lead the discussion around the science, and I'm going to come back and join you shortly to talk about these sort of start to touch on the ethical and legal issues that we need to consider if we're going to advance this area and answer the question of providing a new technology in this space. So Katie, over to you.
Okay, so yes, I have the pleasure of introducing the science behind this. So we're going to start talking a bit about the gametes. So what are gametes? Basically, this describes the reproductive cells of an animal or an organism. So for us, it would be ova or eggs and sperm, and they're derived from a cell called a germ cell. And these usually develop or at least differentiate within the gonads. So for humans, that would be the ovaries, and females are the testes and males. So gametes are special cells in that they're haploid, meaning they only have one copy of each chromosome, and this allows them to fuse during fertilization to complete a diploid cell that has 2 copies of each chromosome. And we're going to be talking a lot about stem cell technologies. So basically, we're talking about cells here, stem cells, which have this amazing ability to self-renew and to also become many other different types of cells. So this is a term called pluripotency, and we use this to describe cells that can become other cells in the body. So this schematic here shows that this can be pluripotent stem cells, or even stem cells that are present within a tissue, which can be activated during repair or regeneration. And you can see the schematic showing, do we have a laser?
Sorry, I don't know about that.
I'm not sure if you guys can see that, but the stem cells' ability to become blood cells, endothelial cells, muscle cells, or neuronal cells. And the applications of these are really widespread. So in the lab, they can be used for cell therapy or regeneration, a huge amount of research going into these cells and their applications for different tissues, and then also drug screening. But this slide is to really point out that stem cells can have different origins. So 2 main origins, especially when we're talking about human stem cells, are embryonic stem cells or induced pluripotent stem cells. And so embryonic stem cells are cells which have been isolated from an embryo, from the blastocyst. So the inner cell mass is removed and grown in a dish. And again, these have that pluripotency where they can be differentiated into all these different types of cells in vitro. iPS cells are slightly different in that these are cells which were somatic cells, so typically fibroblasts or blood cells, and which were taken and put with reprogramming factors and reprogrammed back into pluripotent stem cells. And again, these can be differentiated into many different cell types. So while the function and the use of these are pretty similar, the origin is very different, and there are both legal and ethical implications to the origin, in particular when you're talking about human embryonic stem cells. So in vitro gametogenesis kind of takes these 2 ideas and puts them together. So the idea here being to use pluripotent stem cells to create gametes in a dish. And this scheme, they're showing the use of these induced pluripotent stem cells, which have come from an adult tissue such as fibroblasts or blood, induced into germ cell-like cells, so primordial germ cells. These are the kind of founder population of germ cells. And then these are differentiated into the sperm and oocytes in a dish, which can then be potentially used to create an embryo. So why would we want to do this? So I think from a research perspective, there's a lot to be gained from this, in that it'll allow, probably for the first time, labs to study human gametogenesis or human gonad development, and as a platform to model infertility or other conditions which arise in these cell types. And this has typically been really challenging because these cells arise at almost 2 weeks gestation, so 2 weeks of pregnancy. It's a time that it's near impossible to get these actual cells out of a fetal or an embryonic source. So this provides a platform in the lab to study these cells. But of course, there's been a huge amount of thought and hype around the use of these for assisted reproductive technology. So the ability for people who can't produce gametes or viable gametes to now have genetically related gametes produced in a dish when they wouldn't have been able to otherwise. There's also been a lot of talk about regenerative medicine. So one example might be children who had chemotherapy and who lost fertility potential, but who still have gonads, who still have testes or ovaries, and maybe using something that's made in a dish to replace their fertility potential. And there's also been talk about same-sex couples being able to use this technology to create gametes from each parent, which could then be combined, or even solo parents. I think also in the future, there will be the potential to combine this with gene editing to correct disease-causing mutations where parents, might both carry a mutation that would be passed to an offspring, but that could be corrected in the dish before an embryo is made. And there are massive implications outside of humans for conservation. We know there are groups who are working on IVG in white rhino, and other groups who are looking at even using it as part of a push to de-extinct certain animals, such as the Tasmanian tiger, the mammoth, and the dodo. So I know the slide's really busy, so I'm going to go very slowly, but we're just going to talk a little bit about the development of the gametes. So as I mentioned, the gametes arise from germ cells. So primordial germ cells, or PGCs, these are really the founding germ cell population. And they arise at about 2 weeks in the epiblast, shown here. And they actually migrate through the developing hindgut endoderm and arrive at the gonad, which is also just developed. And that early gonad actually develops the same in both females and males. It's called a bipotential tissue. And at around about the time that those germ cells arrive, the gonad then starts to differentiate either into an ovary if you have 2 X chromosomes, or a testis if you have an X and a Y. So the germ cells come into the ovary in females, and they start to proliferate as oogonia, and they undergo meiosis. So they divide their chromosomes so that they just have one set. They then actually pause for quite a while until birth and then puberty, where then they continue to develop into oocytes. In the male, the germ cells arrive and they proliferate as gonocytes and pre-spermatogonia, and they actually mitotically arrest at that point. And they don't do much until birth, when they become spermatogonia, and then again at puberty. Often these things are brought on by hormones when they undergo meiosis to create the haploid spermatid population. So if you look at this whole process, I just think it's really interesting to point out from their development to their functional development as a haploid gamete, it's about 15 years this process takes. And this is something that people are trying to recapitulate in several weeks or a month in a dish. I also want to point out that although germ cells come from elsewhere in the body and move to the gonad, the gonad is such a complex environment that we just don't fully understand. And it's becoming very clear through all of the research that's been done that this environment is so important for the proper development and differentiation of these cells. And so this is just showing the inside of a testis and a testicular cord, where you have the germ cells which develop into the spermatogonia, into the spermatid. And they do this in really close collaboration with these cells called Sertoli cells, which nurture them and provide them with the environment they need. And this is all closed off from the external environment by a basal lamina. And then outside of that, you have cells producing the important hormones to keep this process going. And another thing that I think we really need to think about as we're developing these methods is this idea of epigenetics and reprogramming. So epigenetics refers to changes that are made on the DNA. So they don't change the DNA code, but they can really change the way the DNA is used. So imprinting or epigenetic marks can turn a gene off, or they can turn a whole chromosome off. But during development of the germ cells, these epigenetic marks get completely wiped so that those paternal imprints are gone. And these germ cells basically arrive naked of any of these imprints, which then get reestablished in the gametes and then wiped again in the early blastocyst. And so again, we don't understand how this works, especially in humans, as these germ cells migrate. And so trying to recapitulate this in a dish is going to be a real challenge. So coming back to the scheme again, when we talk about in vitro gametogenesis and this process of going from iPS cells down to sperm and oocytes, I'm just going to touch a little bit on where the science is at and what people have achieved. So this scheme shows iPS cells going to spermatozoa and all of the steps along the way, including imprinting, differentiation, and meiosis. And so as early as 2008, groups were able to create these primordial germ cells in a dish from iPS cells. And it was found that if you took these and you put them back into a male mouse, they could become sperm within the testes of the male mouse, and those sperm could produce offspring. So they had that germ cell potential. Groups also showed that if you took testes out of a mouse and you reconstituted them in a dish, that these PGCs could become spermatogonia, although they struggled a bit going through meiosis, again showing that that testicular environment is really important. Other groups have used genetic manipulation to get to various stages in this scheme, including meiotic and haploid cells. And so the genetic manipulation here is often using a virus to introduce certain genes which would be massively overexpressed and really push the cells into this fate, although the science behind this is not that well established, and it's not clear whether they're creating really functional gametes. In terms of the ovary, again, if you take a PGC here created in the lab and you put it into a female mouse into their ovaries, they can become eggs that create offspring. And when you culture them with these reconstituted ovaries, they can also become an oogonia, which some of these oogonia will undergo meiosis, but really with poor outcomes and poor fertility potential. And recently, probably the latest advance has really tried to not only create the germ cells in a dish, but create that gonadal environment. So in terms of ovaries, this group has been able to create follicular cells, which then support the germ cells, create these follicle-like structures, which can then give rise to offspring. So really achieving actual IVG in a dish with both the germ cells and the ovarian environment in mice. And if we look at humans, I guess there's been a similar trajectory in terms of the advances. More recently, people have shown that you can create germ cells in a dish from iPS cells, and that if you put these cultured with mouse ovarian cells, you can get rudimentary oocytes when some of these undergo meiosis. There's also been genetic manipulation to drive these into secondary oocytes, again, using viral vectors and overexpression of genes. And also rudimentary sperm have been developed when these germ cells have been taken and cultured with mouse testicular cells. So now I think the field is very much focused on trying to come up with the testicular and the ovarian environment in a dish derived from human cells, because it's not feasible to continue to use mouse cells. And so there have been quite a lot of papers out about creating testicular somatic cells or even organoids, which look like the gonad, and ovarian follicular cells, many of these still in preprint. And then I just wanted to highlight this paper, which again in mice showed that you could take cells from a male who was infertile because they had an extra chromosome. So in this case, 2 X chromosomes and a Y chromosome. And the researchers found that when they created germ cells in a dish, that these germ cells actually spontaneously lost that additional chromosome and therefore could create functional sperm, which could fertilize eggs and create healthy, fertile offspring. However, what wasn't mentioned when this was massively publicized in the media was that almost half of mice in many cases actually developed cancer. Again, you know, a lot of promise without really highlighting the risks of these technologies. And yeah. So I think from a scientific perspective, there are still a lot of barriers to success if we're going to talk about IVG in humans. We still have a really poor understanding of how the gonads or the germ cells or the gametes develop in humans, again, because accessing those tissues from human fetuses or human embryos is really, really challenging, both ethically and also logistically. And we also have a really poor understanding of those non-genetic epigenetic marks and how these occur. And this issue was speeding up such a slow, long process in a lab. We just don't know what kind of effect that might have on the gametes. One of the big things which Megan is going to touch on as well is if we can produce sperm and eggs from human cells in a dish, how do we test that they're good? In mouse, you can create a mouse, you can fuse these to create an embryo, but in humans, the law around this is far more strict. And how do we ensure that they're not damaged along the way? The gonad and the body are so good at protecting the germ cells. When we create these in a dish, you know, there's all sorts of other environmental factors that we need to consider. And I always think it's interesting to think about taking a tissue such as a skin cell, which might have had UV damage, and creating a germ cell out of this. How do we know that that germ cell or that gamete is okay, doesn't have DNA damage, which would cause, you know, something to go wrong in a child which was conceived using it? So I'm going to hand back to Megan.
Thanks, Katie. So I have lots of questions, and I'm going to ask you some questions at the end as well. But I think, as Katie mentioned, I really want to focus now on those particularly around this issue of, if the science is looking promising, when is it ethical to start using the science to achieve some of those objectives that Katie outlined, particularly around helping people perhaps overcome infertility or provide other choices with their families? And I think this is the challenge, not only from an ethical perspective, but also I think legally. So I'm going to kind of start there and say that, as I mentioned earlier, within the stem cell community, we've been thinking a lot about this work. And I just want to draw your attention to a paper that came out in 2021. And this coincided with the revision to the International Society for Stem Cell Research guidelines around stem cell research and clinical translation. Now, these are quite a all-encompassing set of recommendations, but we try to revise these regularly to provide particularly the scientific community, but also others. I know editors of journals look to the recommendations here to assess manuscripts around whether work is within the acceptable boundaries, I suppose, or not. And one of the areas that we touched on in the revisions in 2021, this is the third iteration of this document, was IVG. We explicitly wanted to put some recommendations around what kind of oversight and consideration should researchers comply with, recognizing that we're an international society, and I'll come back to that point in a sec. But as you can see in this sort of illustration, there are different, as colored, ranging from yellow, green, orange to reds. There are different sort of levels of research oversight that we recommend within this document. We recommend that there are some areas of research that do not require institutional and formal review. Others where you may wish to alert or at least discuss with your ethics committee, institutional ethics committee, to ascertain whether there are any local or other considerations. But there's this kind of area in the middle that I sometimes think of as sensitive areas of research, where they require a much higher level of review. So not just your usual institutional ethics review, but a specialized scientific and ethics review. where perhaps the conventional or the standard ethics is supplemented with other experts, but where the technology is confronting on one or more ways. There's also another layer of research that we see as that should be prohibited. And we do now speak a little bit more in the guidelines around areas that should be explicitly prohibited like forever versus areas that perhaps if some scientific challenges are overcome and there is general community support for that work, they should be considered. So that's the structure of the document, or at least the regulatory pathways and recommendations within it. And when it comes to IVG, as is discussed in this paper, it sits at this kind of category 2. And I think that is a really kind of sensible suggestion for all those reasons that Katie just outlined. We know there are so many open questions about whether we can recapitulate in a dish this very, very complex scientific biological process. And it's not as simple as perhaps those headlines I showed earlier imply. And for me, when I think about it, and we haven't yet gone into this much detail in the ICCR guidelines, but I often think about this issue around specific consent. And I know this is raised a lot in other areas, but in the stem cell field, as Katie mentioned, we use a variety of different stem cells. So sometimes the stem cells are from an adult or from a child, or they're obtained from the tissue itself. But in other cases, they are these pluripotent stem cells. So they're either induced pluripotent stem cells, which again could come from a body cell, as Katie mentioned, maybe a blood or a skin cell. But they could also come from embryos. And I think that at least when I reflect on some of the consent forms that I've been involved in, particularly around embryonic stem cell research and iPSCs, the way we describe the stem cell technologies is always using somatic cell examples. So it talks about, do you support your cells being used to grow heart cells or liver cells or kidney cells? But we rarely stipulate it could also grow gametes. And I think there's an inherent assumption there that people who are happy to donate their embryo or who are happy to donate their cells for basic laboratory research are also comfortable with this other use. And so I amended this figure that Katie had used in her slides to again bring up this point around embryonic stem cells as well as iPSCs as a source of these primordial germ cell-like cells. And we might come back to the terminology like cells later in the discussion because I think it's quite interesting. But I think the source matters, as I would argue in many other cases. But I think in this context, I think we should be thinking a bit more about specific consent. And that would also go to how the cells are then used and what sort of animal studies that they're used within. The other obvious issue is, can we make an embryo? And if we do make an embryo, because how do you test? How do you test whether a gamete or gamete-like cell is viable, is functional? The only way to test it is by fertilization and then watching development. And what are the considerations there? What should we be thinking about? And again, does the donor have input, and should they be aware of those restrictions? As Katie mentioned earlier, some of the techniques that have been used to achieve success in mouse studies involve use of surrogates, where you have to get the maturation stage we need, you have to stick the kind of early spermatogonial cell into a host testis. So I can't imagine us doing that in research in man, but maybe some might want to use an animal model. And what does that do to the kind of proposition in terms of consent and donation? And also many other questions. And then, of course, if we do create an embryo, how long should it be cultured for? Should it be consistent with the kind of standard practice in most jurisdictions around the world, which is 14 days post-fertilization? At least it is at the moment, although that is also a point of discussion in the scientific community and elsewhere at the moment. So there's some of the things that I think about. I'll be curious to know and hear from you. We really want to hear some feedback from you about some of the other issues that you're interested in. But I thought it would be helpful now. Oh, I just wanted to go back and say that, just to be very clear, within the ICCR guidelines, when I talked about this kind of regulatory pathway, there's a distinction made around attempts or research that involves in vitro culture of embryos that have been derived from fertilization using IVG gametes versus taking that embryo and putting it into the uterus of an animal or of a human. That is still explicitly prohibited in the ICCR guidelines. And again, I think that for the obvious reasons that Katie has outlined, should very much still stand. So I wanted just to kind of go back to the Australian law, because whilst the ICCR guidelines are international, embedded within them is recognition that no matter what the society, our scientific professional society, may kind of deem to be acceptable or not, the reality is that we work within some kind of regulatory framework around the use of human embryos in research in most jurisdictions around the world. And here in Australia, we've had law in place since 2002. It has been amended twice, but in a kind of nutshell, you can apply for a license to do certain research, and I'll go into that in a little bit more detail in a sec, where the research is assessed, again, by this kind of special scientific and ethics oversight committee. In Australia, it's called the Embryo Research Licensing Committee, and it's a facet of the NHMRC. And what they're looking in the application, what they're, I suppose, evaluating in the application is whether this particular proposed use has a likelihood of significant advancing knowledge for improving technologies used in fertility treatment that can't be achieved by any other means, and also general knowledge around developmental biology and a lot of the, obviously, the work. So I always think it's interesting when we talk about this law, because a lot of people talk about it as the embryo, as a stem cell law, and it's actually not. It's the use of human embryos in research. And I think, you know, the use of human embryos is much broader than just stem cell derivation. But anyway, the other aspects of the application are justification around how many embryos would be deemed necessary to achieve the project goals. Also, a great deal of clarity around how consent would be obtained and from whom, and ensuring that they're not that they're compliant with requirements of under these two pieces of law. And of course, it requires HREC approval independent of the licensing process. So in Australia, we can apply for a license to use an excess IVF embryo. That's an embryo that the original couple or person that the embryo was created for to treat them for infertility has been deemed in excess of their requirements. But as I also kind of alluded to, we also can now, since various amendments to the original 2002 legislation, use other types of embryos, such as those derived from somatic cell nuclear transfer or from mitochondrial donation, which is, of course, the most recent modification associated with May's law. And what remains explicitly banned in Australia is, of course, human cloning, where you'd use the technique of somatic cell nuclear transfer to make an embryo, but then transfer it into the uterus of a animal or person. And that has been, obviously, continues to be prohibited. You can also not create an embryo without a license from the committee I mentioned earlier. You cannot culture an embryo for more than 14 days. You cannot create human-animal chimeras, where chimeras is defined as adding animal cells into a human embryo. Of course, you cannot genetically alter human embryos or gametes. So many of the approaches that Katie outlined in terms of methodology would not be allowed in Australia, even if you were to get a license, because of that restriction. And as I've indicated here, there's been a removal of a previous restriction around embryos with genetic material from more than 2 people. So it now does allow mitochondrial donation. So you've probably heard a lot of that before in terms of mitochondrial donations. I know Catherine and many others have done work in this space, as well as Julian and chimeras. But what I think is pertinent is to look at some of the clauses, and these are just two of the articles under the Acts that explicitly relate to IVG. And as you can see, each have a severe penalty associated with breaching them. And I think, just to be clear, it's always within this law has always been envisaged that you only create an embryo for the creation you only create an embryo for the purpose of achieving a pregnancy, with a few exceptions. And I think that's the fundamental principle within our law. And of course, that poses problems when we want to create an embryo to see whether these gametes or gamete-like cells are viable. So that's the tension that we sit within. And I think within Australia, as you may have followed from some of the discussions around the use of human stem cells to make embryo models, such as the work of Jose Polo and his blastoid work at Monash, I think we'll have to see how the Licence Committee view an application. Perhaps it might view an application for research favourably, so therefore apply a rigorous evaluation to meet those criteria I mentioned earlier, but allow the research. But I'm not 100% sure, and I'd be really curious if anyone on the in the call today has an opinion on that. But I think my instinct would be that we need to see sort of a test case effectively, and it may be possible. What won't be possible is definitely any kind of genetic modification and any kind of extended culture. So stay tuned. I think that will be a really interesting discussion. I wanted to kind of finish the formal part of the presentation by just going back to the opening slide. And, you know, I kind of talked about these earlier around these headlines that really kind of elevate my blood pressure. And partly that is because of the screen grab on the right-hand side of this. And you can see here, this was sent to me by somebody in the community, and it's a company overseas that provides a lot of information about treatment for infertility. There's a matching page for female infertility. Here it's focusing on male. And if you read through quite a lot of information, at the bottom of the page, it talks about the fact that they are using, in this case, the patient's own fat, I believe, and injecting that into the testis. But they use rhetoric and kind of imply that what they're offering is an extension of some of the science that Katie spoke about. And I think you'll see that this is so different. It doesn't involve any of that complex kind of replication of development that Katie was talking about. It's really just using the word stem cell, and it's like a mirage. But unfortunately, I think it is quite a powerful mirage. And you know, I follow this sort of area, this marketing or mismarketing of therapeutic products in many other areas. But infertility, I have to say, I was quite surprised that it initially that it was in this area as well. But it has always been here. And I suppose, like many other conditions, there's people here who will do anything to overcome their infertility, including having fat injected in your body. And again, just to illustrate, I've worked at a series of Australian government-funded stem cell research initiatives, most recently Stem Cells Australia, and I now, as Catherine mentioned, work at the Novo Nordisk Centre for Stem Cell Medicine, but we routinely get inquiries, people asking us, can stem cells help me? And I just wanted to share this quote from someone who contacted us that said, is there any advancement for the treatment of non-obstructive azoospermia? So azoospermia means no sperm in the ejaculate. Is there any chance for a cure with stem cell therapy, fat or bone marrow transplantation into the testis for unexplained infertility? What is the cost of autologous stem cell therapy for the above said conditions? So I think, again, this is a hard illustration of the plight that many are in, not only because of their desires, but I think of the misrepresentation by clinicians and others in the marketplace. So it's something I think we need to keep an eye on and continue to hold to account. So I think we wanted to kind of finish there formally, but then open the floor for conversation. And I don't know about Katie, I'll let Katie answer, but for me, the answer to this question is still that the future is a very long way off. And if we are to advance towards the future, I think we have some significant challenges, both at a scientific, at a societal and a regulatory level. Katie, would you like to make any final comments before I stop sharing?
I would agree with that. I mean, I think that it'll come probably more quickly than what we're prepared to accept, in particular with the buy-in of so many large pharmaceutical bio startups. But yes, it remains to be seen how safe and effective it will be when it first comes out.
And how we'll test that.
That's right.
So I'll just flash up. We have a couple of papers here that hopefully you can look back at the recording to gather if you wanted to read some more. But I'm going to stop sharing. And as I said, we'd be delighted to have any questions.